Aloha mai kako, o kiki ko'u inoa, and buenas and hafide ina anhu si kiki Masga. And I will be presenting on the distribution, diversity, and density of plant pathogens in the ahupuaʻa of Kailua. We are extremely grateful to stand here on this ʻāina of Kawainui, which was once a rich agricultural region. We stand here next to Ulupoheao, which is a sacred site containing pohaku from all across Oahu. And the name Ulupo, as Uncle Kaleo was sharing, expresses how rich of an agricultural region this once was, that the forests were so rich that the understory seemed like it was nighttime. But if you look around us now, we can see that we lost a lot of the agricultural region. However, we were welcomed today by the stewards of this ʻāina, who are working towards ʻāina restoration to restore Kawainui into a fish pond again and restore the Kalo patches. Now, as we know, in an ahupuaʻa system, streams run from mauka to makai, and specifically for Kailua, the streams run from the source of Pikoakea from mauka, here through Kawainui to mauka in Kailua Bay. And here in Kawainui, we have Kalo patches being grown. And unfortunately, Kalo is prone to contract diseases that result in root rot and leaf blight. And we attribute those diseases to plant pathogens. And plant pathogens are introduced when plants are introduced from other regions, and they bring along with them plant pathogens and pests. And with rain and wind, those plant pathogens are washed into the streams that feed Kawainui. And here in Kawainui, they use an ʻaoi system, otherwise known as flood irrigation. And plant pathogens may be introduced upstream, and they result in rotten kalo and lo'i that are unable to be used for agriculture. And it's important to understand where these pathogens are present and how they are introduced, so that we may prevent them from infecting the lo'i and reach our goal to produce happy kalo. We focused on a specific root rot pathogen of Pseudomonas aeruginosa. It's a gram-negative bacteria with flagella, which promotes motility in water. It develops biofilms that resist antibiotics and competition, but is generally abundant in soil. However, it has the potential to develop root rot in kalo. So we began experimental design by identifying who the plant pathogens are first and where they are located. We did that first by sampling all across the water streams in Kailua from mauka to makai, and here you can see the map and the points where we collected water. We collected 1 liter samples in duplicate from the streams, and here we have pictures of my cohort collecting samples at the beginning of the summer. We then moved on to DNA extraction, and that is basically filtering the water onto a pore filter that traps the bacteria. We performed lysis to break it down and used specific reagents to precipitate out the DNA. For identification, we paired the DNA with the 16S primer, which is specific to bacteria, and we amplified it to send off for 16S sequencing. And we hypothesized that there would be a decrease of plant pathogen abundance as the water went from mauka to makai. So we expected to see high plant pathogen abundance before Kawainui and more after, and less after and in the bay. And here is a figure that we have. So to orient you to this figure, on the y-axis we have the relative abundance. So this is basically like the percentage, and this is on the scale of 1. So this is specifically for plant pathogens relative to the whole bacterial community. On the x-axis, we have the ahupuaʻa locations separated into bins of before Kawainui, in Kawainui, after Kawainui, and in Kailua Bay. And these bins are separated further into, you can see the different colors, and those are separated into the bacteria to the genus level. And we can see that before Kawainui, plant pathogens take up roughly 4%. In Kawainui, they take up roughly 2.5. After Kawainui, they take up 0.05. And in Kailua Bay, they take up roughly 1.5%. And we wanted to focus on these three genuses just in case you guys had something pointing out on these figures. So we can see here that Hydrogenophaga is the most abundant bacteria, and that's because Hydrogenophaga is commonly found in wastewater, in ponds, and source water. So it's not a surprise that we see it so abundant in the water, as water is its normal habitat. And as we predicted earlier, we see Pseudomonas here. We can see it in large abundance before Kawainui and in Kawainui, but we don't see it as abundant after Kawainui and in Kailua Bay. And we also wanted to point out Vibrio, because you can see here in Kailua Bay, there's a large genus that's not present in other bins. And that is because Vibrio is a common marine bacteria, which is why we see it in such high abundance in Kailua Bay. And through this figure, we can assume that Kawainui wetland microbes change. So the microbes that are not plant pathogens basically increase in abundance, which is why we see such different decreases as the water runs from Mauka to makai. And then next, we want to investigate how many of a pathogen are present at the specific sites. And we hypothesize that there would be low abundance of P. aeruginosa before Kawainui, higher in Kawainui, and less after. We did that by pairing the DNA with two different primers, one for 16S to quantify the whole bacterial community, and one for O-acetylase to quantify Pseudomonas aeruginosa, along with cybergreen, and we ran that through qPCR. And here's a figure of the bacterial quantity, and this is on the log scale, so this is the log copies. And we can see here that there's an increase as the water runs from before Kawainui through Kawainui to after, and in Kailua Bay, it kind of steadies out. And for Pseudomonas aeruginosa, we can see that there are low quantities before Kawainui, while there are higher quantities in Kawainui and after Kawainui, but they decrease again here in Kailua Bay. And for the whole bacterial community, we say that Kawainui increases microbe quantity. So because of the slow-moving waters here in Kawainui and after Kawainui, the bacteria may remain in the slow-moving water and multiply, which is why the quantities increase. And for the same reason, P. aeruginosa accumulates in the water in Kawainui and after Kawainui because of the residence time in the water. And for future work, we would need to quantify the amount of Pseudomonas aeruginosa in rotten Kalo roots to determine the effect of Pseudomonas aeruginosa on Kalo growth. And I want to wrap this up by bringing up the mo'olelo of Makale. So in the mo'olelo, we know that the stick of Makale was used to bring the fish from Kawainui through Maunawili up to Hawaii. And this mo'olelo reminds us of the connectivity of the water streams from Mauka to makai. And it's important to know that because we must treat all the water from Mauka to makai well, so we don't introduce bacteria, so we can reach our goal to have happy Kalo that can directly support the community. I would like to thank Kumu Kiana and Kumu Matt, as well as the members of Lab Pio Frank and the stewards of this land, Kauluakalana. This research was sponsored by the University of Hawaii at Manoa and supported by the National Science Foundation. Thank you. Are there any questions?